Okay, good afternoon, everyone. My name is Tun Naw, and I will be presenting my research on supported lipid bilayer formation from bicellular mixtures. So before I present my research proper, I would like to talk a little bit about the PhD program that I'm in. I'm in this program called NTU SKQ Dual PhD program. So it's a joint PhD program between NTU Singapore and SKQ Korea that's designed to promote transboundary research and strong partnerships between the two universities. So under this program, students have to spend half of their PhD at each university, and they do their research under two supervisors, one at each university. So I have already completed my first half of the PhD at NTU Singapore, and now I'm in SKQ Korea to complete the second half of my PhD. And I'm also enjoying my time here with lots of good Korean food. Okay, so now to my research. Okay. Okay, so I'm making supported lipid bilayer platforms, SRVs in short, that mimic the basic architecture of the biological cell membranes, which is the lipid bilayer. So SRVs serve as model platforms for fundamental membrane studies and applications such as biosensors. SRVs are formed when the lipids are deposited onto solid supports here, and they are physically stable because they are formed on solid supports, and they are mobile because there is a water layer between the lipid bilayer and the solid support. So the lipid molecules can move freely within the plane of SRV and also laterally along the surface, just like the actual cell membrane. So these properties make SRVs very suitable for a wide range of surface sensitive techniques. Next slide is okay. So there are lots of techniques to fabricate SRV. The most popular one is vascular fusion, where the small spherical vesicles absorb and fuse on the solid support to form SRV. So it is fast and it's simple in terms of procedure, because you just need to deposit the vesicles onto the substrate and they will absorb and then rupture to form SRV. However, the vesicle preparation is not so straightforward, because the main challenge is to get the uniform size distribution among the vesicles, among the very small vesicles. And SRV formation is also limited by many parameters. For example, if you put cholesterol in the vesicles, you cannot get a complete SRV formation because cholesterol hinders the vesicle deformation. Okay, so to overcome some of these limitations, and recently an alternative SRV fabrication strategy has been tried out using bicells. So bicells are mixtures of long chain lipid and short chain phospholipids. So when these two types of lipids are mixed in an aqueous solution, they self-assembly into a bicell. So these long chain lipids will self-assembly into a planar bilayer, and then the short chain lipids will form a rim around the edge of the bilayer. So so based on this Q ratio, which is the ratio between the long chain lipids and the concentration of the long chain lipids to the short chain lipids, and also other parameters like temperature and composition, bicells can take on many other different forms other than this common disc shape. So the one main advantage is that the sample preparation is minimized, by which I mean there's no need for well control of size distribution, which is the main challenge in vesicle preparation. And another thing is that SRB formation is energetically more favorable because bicells contain the planar region, which has larger contact area with the substrate than the vesicles, so less adhesion energy is required for bicell absorption and subsequent SRB formation. So as of now, the application of bicells in SRB fabrication is still very new, and in the literature, only a few studies have reported the use of bicells to fabricate SRBs. And my research is to further explore the bicell-based SRB fabrication under different experimental conditions to identify the optimal conditions and to have a better understanding of the bicell-based SRB formation mechanism. So there are a few, okay, so these are the experimental methods I use to monitor the kinetics of bicell adsorption and SRB formation. I use quartz crystal microbalance with dissipation, QCMD, that characterizes the mass and viscoelastic properties of the adsorbed lipid layers by measuring the frequency and dissipation shifts. So the frequency shifts indicate the mass absorption, and the dissipation shifts tells you whether the adsorbed layer is soft or rigid by measuring the energy loss during one cycle. So for SRB, the energy dissipation is quite low, and so to visualize the structure and formation of SRB, I use fluorescent microscopy and fluorescence recovery after photo bleaching FRAP techniques. These techniques also tells you the lateral diffusion of the lipids in the SRB. Okay, so this is the mechanism of SRB formation from bicels. First, bicels in solution approach the substrate, and then they adsorb. And after the bicels cover the entire substrate, they will fuse, and SRB will form. So when SRB is formed, the short chain lipids will leave the surface because they have higher solubility than the long chain phospholipids, and then the long chain phospholipids will remain on the surface to form SRB. So the corresponding readouts of these SRB formation stages in QCMD and in fluorescent microscopy are shown here. So so one thing to know is that when SRB is formed in QCMD, the final frequency shift will be around -25 to 30 Hertz, and then the dissipation will be around 1 or less than 0.5 if the SRB quality is very good. And I first looked at the effect of NaCl concentration, salt concentration on SRB formation from bicels that are prepared in 150 millimolar NaCl buffer, and then compared with identically prepared vesicles without DHPC lipids. So the test range was from 0 to 500 millimolar NaCl. So in the case of bicels, NaCl concentration does not affect SRB formation from SRB formation. Even in the absence of NaCl, bicels still form SRB. And on the other hand, SRB formation from vesicles is affected by the NaCl concentration, and so there's no SRB formed in 0 and 25 millimolar NaCl for vesicles, and from 50 millimolar NaCl onwards, SRB formation occurred. So so these dotted lines represent the SRB values, and so okay, so this is the comparison of the SRB quality as seen by the fluorescent microscopy and the FRAP technique in 150 millimolar NaCl buffer. So SRB formed from bicel has higher quality as shown by the uniform fluorescence than the SRB formed from vesicles where they are unruptured vesicles, sorry, as shown by the bright spots here. So these are the unruptured vesicles. And next, I looked at the effect of bicel surface charge on SRB formation on different oxide substrates. As a control, I checked the absorption of zwitter ionic bicels, which are electrically neutral, onto SiO2 and TiO2 surfaces, which are both negative at physiological pH of 7.5, and aluminum oxide, which is positive at pH 7.5. So the results show that these zwitter ionic bicels form SRB on silicon oxide only, and there was very little or no absorption on TiO2, and there's some absorption on aluminum oxide. And with positively charged bicels that are prepared by adding DOEPC lipid, which are positively charged, and the SRB formation occur on SiO2. With positively charged lipids, and with negatively charged lipid that are formed by adding different fractions of DOPS, there was no SRB formation. There was only absorption, and in some cases there's minimum absorption. So And on titanium oxide, the same results as in the as on the silicon dioxide, so which is also a negative substrate. And for aluminum oxide, the trend is opposite as expected, since the aluminum oxide is the positively charged. So all the negatively charged biser form SRB on aluminum oxide. And so next, I explored the SRB formation from bisers made of DOPC and LA, which is the lauric acid, which is a fatty acid. So the main motivation behind this is to see whether SRB can still form when the synthetic short chain lipid, DHPC, is replaced by the fatty acids, the lauric acid, which is the naturally occurring compound, a surfactant actually. So the results show that SRB formation is possible under certain conditions, like Q equal to 0.25 and Q equal to 2.5. So in summary, SRB can be formed from bisers regardless of salt concentration in the bulk, and biser surface charge can be fine-tuned to fabricate SRBs on different oxide surfaces with different charges. And lastly, biser composition can be engineered for broader SRB fabrication as well as application scope. So biser methods have proven to be robust and versatile. So that's the end of my presentation. Thank you for listening and have a great day.